to talk about is a bit about the value and the dynamics of dead and dying trees. Then I want to tell you a little story, a story of some research that I've done looking at the response of some mammals in Oregon to forest fragmentation. Now I want to say at the beginning that this is very different than what's happening on Mount Jumbo. But I still think it's useful because it's kind of an analog of forest processes. And it tells us some things that are going to be shared, that is going to be similar on Mount Jumbo in the forest. And it also makes us think about some things that will be different in Oregon, and that will be different in a situation where a patch of forest gets carved out of a much larger continuous tract of forest. That's different than what we have on Mount Jumbo, as Paul talked about. But thinking about these things will help us get some ideas and get the old gears turning for thinking about processes in the forest on Mount Jumbo. And then, just to keep me honest, that's why I have part 3, to remind us that we don't want to just leave the processes that happened in Oregon. It's really fun what happened in Oregon. Really interesting, really an exciting story, the way that small mammals respond to forest fragmentation. But we don't want to leave it there. We do want to come back to Mount Jumbo and say, okay, how are things different or the same in the forest in Mount Jumbo? Given that it's a different place, different species, and a different scene in the sense that these patches of forest on Mount Jumbo aren't just carved out of a big continuous patch. They're sort of arisen from this lack of disturbance that Paul talked about. Okay, so first of all, let's then think about the value and the dynamics of dead and dying trees. So again, I'm not going to talk about all the cool, beautiful things of big, fat, fluffy, intact trees. I want to talk about dead ones. And I'm going to say that, and we'll talk about this more on Saturday, for a lot of wildlife, the tree isn't really all that great until it does get sort of beat up. Broken top trees, that when the tops get broken out of big trees, that's when you see a lot of use by all different kinds of owl species, flying squirrels, sometimes red squirrels, lots of different species use trees that are broken up. So live trees are of course useful, and of course live trees are important. But again, I'm fighting from the underdog tonight and talking about the advantages of the big ones, or the advantages of the dead ones. This is just a big list that you've seen before, listing cavity nesting birds commonly censused in temperate forest ecosystems. The point is just to show you that there's lots of birds that live in cavities. Cavities being defined, different kinds of cavities, of course. Some cavities come from busted up live trees. Lots of cavities come from snags. So that's why I say we've got to think about what happens when dead trees, what's the dynamics of dead trees becoming usable snags? Well, what is the difference between just a dead tree and a snag?
Snake is in branches.
Yeah, it doesn't have branches, and what else?
Bark.
Bark.
Yeah.
Yeah. And so, let's think about why that's good. So, why does that make it better for certain wildlife species?
They can get to bugs easier.
They can get to bugs easier. Yeah. Maybe it's softer too, so it's easier to dig a hole in it to put your babies in. What about the bark? What's the advantage of losing that bark to the forest ecosystem? How does the forest? Yeah, compost. It's sending its nutrients. That tree's been hogging those nutrients its whole life. When it sloughs its bark, it's giving the nutrients back to the soil. And doing what else? What else does bark do?
Mulching.
Mulching, yeah. So that's good for plants. But don't forget the animals.
Food source.
Little houses,
Oh,
right?
places to hide.
Bark can create cover for
Protection.
different critters. Yeah, yeah. So trees lose their bark. That transfers nutrients to the forest floor, and it creates habitat for both cavity nesters on the tree, now snag, and for critters on the forest floor. So how long does this take? Would you guess?
Under 50 years. Between 5 and 10 years for the bark to fall off.
Okay.
Oh, yeah.
Yeah, that's right. So I think we're going to get to your answer in a minute. Yeah, so roughly 6 years. I mean, of course, there's tons of variation in this, but roughly 6 years for a tree to become a good snag. That is, to dump that old bark and to soften up some and lend itself to being made into cavities. It could be a lot faster, of course, if there's critters helping it, like what, woodpeckers, that will within a year get rid of bark, create cavities, those sorts of things. But roughly 6 years, these kinds of processes can take. And then, you know, as a ballpark rule of thumb, and this actually comes from there was a huge synthesis in an ecological journal about the dynamics of decaying wood, and so they talk all about the different ranges of variation. But ballpark, 5 to 20 years, a snag starts to fall apart. Now, of course, you know that a lot of times trees will bite the dust, hit the ground before they become rotten. And so that's the other kind of decadence that I want to think about a little bit. And I'm going to talk a lot more about that in just a minute when I tell you about the Oregon story. Once a tree becomes a log on the forest floor, then what happens to it? How does it process itself to provide more services to the forest ecosystem at that point? What are some of the changes it undergoes? It rots. Yeah. So again, it gets softer, just like the tree did. What else?
The animals are going to start help deteriorating it.
The animals are going to help deteriorate it. Yep.
It holds moisture, it gets hollow.
Yeah, holds moisture and gets hollow. Yeah. So that's right. So that it starts holding moisture as it rots.
Provides places for new things to grow.
Yeah, certain things start to grow. Okay. So as certain things start to grow, that changes not only the moisture, but it also changes the nutrient. As we know, wood's not a very useful thing to eat. Things that eat it, beavers and termites, have remarkable digestive systems in order to process it. I mean, lots of funky symbiotic enzymes and bacteria, protozoans that help them to digest through that cellulose and get nutrients out. But with wood, as it starts to rot, as these fungi, insects, bacteria, other animals start to get into it and burrow around, they leave behind a lot of nutrients. So as wood rots, it becomes more of a source of nutrients. So you can actually measure a log from when it first falls and hits the forest floor, and you can watch over time as this log becomes much richer in nutrients. I mean, all the important nutrients, carbon, you get 2 to 6 times more carbon, nitrogen, phosphorus, not carbon, carbon's there of course, but nitrogen, phosphorus, and potassium that gets produced by these fungi working in that sort of thing. So what's the timeline for this? The decay rate, 100 to 150 years for a log to turn pretty punky. 200 to 250 years is a ballpark for a log to become completely decayed and go back to soil. And well, that's sort of wrapping up a lot of different variations sort of for the Pacific Northwest. Yeah. And when we go out on Saturday, I'm going to take out some sheets, some decayed log data sheets, so that you can poke your pencil into some logs and just sort of get a feel for how people measure different levels of decay in different logs. We'll tear them apart and celebrate their decay. Okay, and then the last thing I wanted to talk about just briefly was this idea of this moisture, that not only is it holding more moisture because rotten wood becomes more spongy, but also you have this respiration going on of all these little organisms munching around in the woods. And as we know, cellular respiration releases H2O as part of the whole process. So that's another reason why rotten wood becomes a reservoir for moisture, which can be really important, especially in dry areas, like Paula talked about here. You know, a place like here in Montana, right on the zone sort of aridity, And so a lot of times, just a little bit extra moisture in a forest under a rotten log can make a huge difference for lots of different critters. Okay?
I want to check an observation. When I think about the logs I've seen in Montana, not Oregon, it seems like on south-facing slopes, you can have a log go all the way through the entire decay process till it's totally flat on the ground, and there's not anything growing there still. It's just little tiny bits of crumbled up wood on the ground.
Yeah.
On a north-facing slope, you may see lichens start pretty quick. You
Yeah.
get a lot more fungi growing and plants starting to grow out of it when it's still above the surface of the ground.
Yeah.
So that
Yeah, that's a good
aspect
observation.
makes a huge difference.
It does, and so what you're seeing there is essentially a situation where the poor log is just being overwhelmed by abiotic factors. I mean, it's just too dry. There's a little bit of a reservoir that a log can offer, that a rotting log can offer in terms of moisture held, moisture retained, and also moisture liberated through respiration. Just isn't enough to make a difference. I just came back from Olympic National Park, where I've got a graduate student who's doing some work that sort of follows up on some of the aspects that I'm getting ready to talk about. And he's studying, he's looking at edges, forest edges, and going out into the clear cuts. And when we were walking around in the interior forest, it's just as you said, if you've ever been to Western Washington, you know the logs there are humongous. They have lots of things growing on them in the forest. But in the clear cuts, they're almost petrified. The logs are. I mean, the logs are there. The particular clear cut wasn't burned, but they're humongous logs, but they're just dry as bones out there. And you realize once you go out there, you check twice to make sure you've got your water bottle, because you think, I'll never come out of here alive. It's so hot and dry in that clear cut. So yeah, I mean, in those cases, the moisture just isn't enough to provide that little microclimate that's needed. So with these kind of sort of dynamics in mind, I want to go ahead and jump you into this story of what happens to certain species when their environment changes from a forest to a clear cut. So this is the creation of drastic edges. I guess I'll just dive right in here. I don't know if that's going to be too dark or not. Can you all see it with the lights on, or?
No, turn the
No?
lights off.
Turn the lights off? Okay. So I worked for a couple of years as a research biologist at Olympic National Park, and you cannot work or be in Olympic National Park without being struck by the issues related to what happens when wildlife populations go from being large and continuous to being fragmented. This is one of those classic pictures that you see a lot, the same kind of picture for Yellowstone. This is one of those classic pictures where you can ask, where does the national park boundary end and the state and Forest Service lands begin? You know, it's pretty clearly, I mean, this is exactly the park boundary for Olympic National Park right here. And this all used to be humongous 8-foot, 10, 12-foot diameter lowland temperate rainforest trees that have been pretty drastically clear cut. If you've ever seen it, it's pretty striking. And so this really got me to thinking, working in this park, thinking about, well, what really would we expect to happen? What are the consequences of this kind of rapid adjustment? What are the consequences to the wildlife populations that are still in the forest when the forest becomes quickly surrounded by unforest, quickly surrounded by clear cuts? So up to this, so what I did was I ended up doing this research in Southwest Oregon, where there's lots of clear cuts. And the question was, what happens when we go from a patch of continuous forest like this to a situation where forests are just little islands in a matrix or a sea of highly modified landscape? So this is just to kind of give you an idea of some of the sites. And what I decided to do is look at the extreme of forest fragmentation. We can come back to this at the end. I wanted to look at situations where patches of forest were completely surrounded by highly modified landscape. So this just gives you an idea of some of them. They were all in Oregon. They were different sizes, but they were all pretty small. They were all in southwest Oregon. There's 13 of them and 5 control sites. It was harder to find control sites unfragmented than it was to find these remnants. But what I thought I'd do is, I'd never studied small mammals before. I'd studied these big glorious animals like coyotes and pronghorn and goats and stuff like that. But I thought, well, if we really want to learn something about forest fragmentation, we might learn a lot more if we look at small critters on small patches. We're wanting to learn processes here about the response of forest wildlife. Small critters on small patches might be easier to do and more productive in a relatively short 3-year study than huge critters on huge patches. So what I did was I used live trapping for small mammals. I never caught any animals, and I never could figure out why. Just kidding. This trap was found by a bear. And in fact, one day a bear came along and found a lot of the traps, and the bear found this little package to be crunchy on the outside and chewy on the inside, and it went along and ate all my animals that day. But usually the traps I used didn't have this hole in them. Got doors on either end, so the animals are caught alive. And the hero of the story here, whoops, I'm not yet there. Here I just wanted to say that what I did was to set up traps in the forest remnant, and then in the clear cut surrounding the remnant, and then also in control areas, nearby areas that were huge areas that weren't fragmented, to kind of get the idea for the dynamics of the system. Yeah, here am I supposed to say, aw. This is old RB01. This is a California redback vole. The genus is Clepthrionomus and the species is Californicus. A species very similar to this one does live up here on Mount Jumbo and here in Montana, Clepthrionomus gapperi. The remarkable thing about California redback voles is, besides the fact that they're just so adorable, is that they're exquisite in their diet. 90% of their diet is truffles, the fruiting bodies of raised mycorrhizal fungi. So they're pretty dependent on truffles because, as we'll see later on, truffles don't do well in clear cuts. And so I picked this species intentionally thinking that this species will be, if there's going to be a response to forest fragmentation, this would be a good species to look at. This particular vole has a radio collar on it. You can barely see the radio collar. And we'll talk a little bit more about that in a little while too. So, the idea was to see what happened when this forest was modified. And what I want to do is just walk you through the results here. So what we have here, this is basically just the number of voles. You can think of this as the number of voles. Here's the number captured in the clear cut, and here's the number captured in the remnant. But within the remnant, we had some traps that were near the edge of the remnant, right, on the outside edge of the remnant, and some that were in the interior of the remnant. So here, all of these traps were 0 to 15 meters from the forest clear-cut edge, and all these traps were 46 to 90 meters interior, in the interior of the remnants. The numbers above the bar are just the number of remnants, and the rest of it's just statistical mumbo-jumbo. But the main thing to take home from this is that there was that these voles were having a real serious negative response to this kind of forest fragmentation. I only caught 3 voles in the clear cut over two summers and about 11,000 trap nights. Only caught 3 redback voles in the clear cut. They don't like clear cuts. On the remnants, there was this significant negative edge effect. So voles were 4 times less likely to be caught on the edge than they were in the interior. Okay, so then this was kind of striking, because for a long time wildlife biologists had sort of propagated this idea based largely on game animals that edges were what? Good, right? Deer do well on edges. Quail do well on edges. Pheasants do well on edges. Lots of critters that we think about, especially those that are harvested and also those that are exotic, do really well at these interfaces. Essentially these sort of ecotones like Paul was talking about. So, but this was sort of some of the first evidence from mammals that there can be negative edge effects as well. So I want to stress, and I'll come back to this at the end, that edge effects aren't always bad, but this tells us that sometimes the edges that are artificially created by human actions can be bad. But this leads us to an interesting prediction. So if we think of a donut, and we think that the interior of the donut, the juicy strawberry-filled part, is better than the outside edge of the donut, then that tells us that if there's a negative edge effect like this, that the density of animals on small remnants would be larger or smaller than the density of animals on large patches. The number of animals per unit area would be what? Would be smaller on small patches, because a higher proportion of the donut is edge, right? A lower proportion is interior. So we can plot that out with these data and find that in fact that's true. That if we look at the number of voles per unit area, say per square foot, that it increases with some scatter around it, but it increases as the size of the remnant increases. So these are tiny remnants, these are bigger remnants, patches of forest surrounded by clear cuts, and in general, the density, the number of animals per square foot or whatever, tends to increase as the size of the remnant gets bigger. So that's important because that tells you that when you fly over these things in an airplane, when you look at them on an aerial map, that the size of the remnant doesn't necessarily tell you the density of all the animals on it. You can't assume that all remnants are created equal. You cannot assume that the distribution of animals is the same across all those remnants. So that's an important take-home message. So that was cool, and that was pretty striking. But there's still a lot more to the story, both ecologically and in terms of management. So now we know there's a negative edge effect on red-backed voles, and we know that that means that remnants, smaller remnants, will have fewer of them. But we still don't know why. So why is it? Well, the thing about red-backed voles is that, as I said, they eat almost entirely truffles. And so that made us think that, well, maybe. Oh, and the other thing about red-backed voles that I have to tell to make the detective work kind of to sort of lay out the detective work that was involved was that people had said that red-backed voles had been caught when they catch voles. The old naturalists that have gone out, when they catch voles, they tend to catch them more in underneath logs than they did not underneath logs. So this made us think two things. Well, maybe truffles are playing a role, and maybe logs are playing a role. So to get at this question, we had to radio collar voles. Now this picture is because Marlin Perkins and all those guys on Wild Kingdom are always wrestling these giant carnivores, wolves and bears and elephants, and people think, oh, that's the exciting stuff in wildlife biology. But I want you to know it takes 3 people to radio collar a red-backed vole. You can see it. There's 6 hands here. It takes 2 people to hold, and they're not hurt. Nobody ever died. They're not hurt from this. It takes 2 people to hold the vole still, 2 people to knock it out. This has a isoflurane in it that knocks it out, and then 2 people to attach the radio collar to the red-backed vole. So it's serious stuff. It's real Marlin Perkins style things. Of course, I was just standing here taking the picture. I had 3 gems doing all the dangerous stuff. Okay, so we put radio collars on to see what roles logs and truffles play. So this, you don't need any statistics to figure out this one. This is a home range of an average red-backed vole. The big fat lines are down logs, pieces of bark, down logs. The numbers indicate multiple relocations. The circles are radio telemetry relocations. And it doesn't take statistics to convince you that red-backed voles like logs. You can see every location is underneath a log. In fact, even though these logs on average only took up about 7% of the collective vole home range area, 98% of the vole locations were underneath logs. Okay, so if voles love logs, then maybe that's the answer to our story. Maybe logs there's more of them in the interior, less of them on the edge, and that's the reason we see this vole edge effect, right? Makes sense? End of story. So we went out there to every single one of our remnants and we put down line transects and we measured the biomass of logs close to the edge and in the interior. Holy cow, what did we find? Exactly the opposite trend than we would have expected if it was logs per se. So what we found is the same kind of graph. Close to the edge, the highest number or volume of logs. Interior, fewer logs. So again, the statistics don't really matter here because there's a trend in exactly the opposite direction. But the main thing here is that if it was logs per se that caused this edge effect, then we'd expect these log bars to go the same way that the vole bars went. But they don't. They go kind of the opposite way. So that's really surprising at first if you expect logs per se, logs alone, to explain the distribution of redback voles. But when you sit and think about it some more, maybe some of you have figured this out, why is this graph not very surprising? If you don't have a vole-centric view, why is this graph, why would you say, well, yeah, of course, that's what I expect to see.
More windthrow and disturbances on the edges.
Yeah, yeah. Think about what happens. When a remnant gets surrounded by clear cuts, what two things happen? Before you get to the windthrow, what happens? Trees get cut down,
Right.
right? Some of them fall into the remnant. They bust up, they're not worth cutting out. So you get a lot of recently fallen logs from the cutting itself, and then after the cutting, you get windthrow. You get trees that are on the edge are a lot more susceptible to being blown down. So what you end up with is a high biomass of recently fallen, coarse woody debris. Recently fallen, that's important. And the reason it's important, I'll show you now. The reason we really realized it was important was because when we really analyzed the data for the redback voles, here's what we found. What we asked was, for all of the logs that were on the vole home ranges, so how decayed were they? So this is basically the number of logs that were recently fallen, just with all their branches intact and in real good shape. And these are really decayed ones, like almost piles of dirt. So the open bars are the logs that were on the vole home ranges. So you can see there's sort of a bell-shaped distribution of the decay classes of logs on vole home ranges. But if you ask which logs did the voles use, that's the white bars. You can see they really preferentially use the more highly decayed logs. So they're not using the logs in the same proportion that they're available. They're choosing rotten logs. Okay, so the plot thickens. So now it's not just down logs per se, and edges, turns out, have high biomass of logs on the edges, but it's not very rotten. So this is kind of interesting. So now we need to bring in the other player, the truffles. Ask what role do truffles play in this whole story? Oh yeah. So, and actually, as my prop, it's dark in here, but I'll pass around. This is a book of truffles. And so you can, for those that haven't seen truffles, you can look at the photographs. Yeah, so, well, somebody say, aren't they like a mushroom?
Yeah.
Yeah, so what a truffle is, that's good. What a truffle is the fruiting body of mycorrhizal fungi that's underground. So you're exactly right, that a mushroom is just the fruiting body of a mycorrhizal fungi that fruits above ground. And truffles are fruiting bodies that are below ground. So you can almost think of them as underground mushrooms. But what's the problem with fruiting underground? What's the disadvantage you have that a mushroom doesn't have? What's that?
Can't find it.
Well, yeah, that's right. That's the human problem, isn't it? Yeah, that's right. It's a lot harder to find. But from the point of view of the fungi, of the mycorrhizae, what's the problem it's got to deal with?
It's spread, it's sort.
Yeah, how's it going to get spread? It can't be spread by wind. It can't be spread by rain. So the only way the below ground truffles, the below ground fungi, sporocarps can be spread is to be eaten by an animal and then be pooped out somewhere else. Okay, so what we did was we studied we looked at some of the remnants, and we looked at, and we put some plots in forests, and you're right, truffles are really hard to find. It takes a lot of work, but we put plots in the forest, and then we put plots out in the clear cuts, and we asked how are the truffles distributed across the clear cut and across the edges. And what we found was that, this is just to sort of review, voles are absent from clear cuts, less dense at edge, and voles are found underneath logs, especially decayed logs. That's the information we had going into the truffle part. And then look, lo and behold, what we found. That if we just look at the biomass of truffles in the forest compared to the clear cut, there are almost no truffles in the clear cut. In fact, we found only one truffle in all of the 80 plots, and guess where it was? Underneath a monstrous Douglas fir that had never burned. Yeah.
They don't take disturbance.
Well,
They don't like disturbance
yeah, but why would that be?
because they're underground, so if you're taking down trees, you're uprooting the ground, and they're
Okay.
probably within the first layer.
But what else? What is it? How does mycorrhizae that you learned last week, how does the mycorrhizae that the truffle comes from make its living?
By living off of the roots of other plants.
Right. And Ray talked about mycorrhizae in knapweed, but it turns out that most of the truffles that you see are mycorrhizally hooked up to conifer trees. And in fact, conifer trees wouldn't grow out there if it wasn't for the mycorrhizae. Foresters are really learning that right now. Now meaning the last 5 or 10 years, foresters are spending a lot of time and money figuring out how to inoculate trees that they plant with mycorrhizae, because if they don't, the trees just don't grow, they die. So that's right. So when the trees get removed, then the mycorrhizae don't have their source anymore for photosynthate that trees typically provide, and so the mycorrhizae can't reproduce anymore, so there's no more truffles. So it wasn't surprising, really, if you know the natural history of truffles, but this hadn't been shown before, that clear cuts don't have any truffles. And then the other interesting thing that we found was that truffles like logs. Truffles were more likely to be found, the biomass of truffles underneath logs was much higher than the biomass of truffles not underneath logs. So logs, you're more likely to find a truffle under a log than you are not under a log. Okay, so truffles in, absent from clear cuts, and also, and I don't have the, I guess you just have to believe me, I didn't bring the overhead to show you this, but turns out that when we looked at the edge effect of truffles, that there were 0 truffles in the 0 to 15 meter edge class. So just have to trust me on that one. That the truffles were absent from the clear cut. There were plenty of them in the remnants. So truffles absent from clear cuts, absent from the edge, more likely under logs than not under logs. So now we're getting somewhere. Now we're kind of getting into some, whoa, she warned me of that.
It doesn't matter because it's not working, so
Oh.
I'm using the one on the camera.
Okay. So now we're getting close to a mechanism of all this business. So truffles then seem to relate to where the voles are found. But now we need to ask, why are the truffles where they are? So, I mean, there's all these fun, different layers of why that you can ask whenever you do a study like this. So, we can look at percent soil moisture. And again, in the clear cut and near the edge and in the interior, and we see a little bit of a trend that clear cuts have less moisture, the interior of the remnants have more moisture. Now this has been shown by lots of people who have done really good studies. We know that interiors of remnants tend to have more moisture. We know that edges have less moisture. They're hotter. They get more solar radiation. They get more wind. They get higher temperatures. This is just a little bit more evidence for that that same story. That's sort of a funny story behind this. I had a bunch of undergraduates who did their senior thesis project with me, but one of them was doing this stuff, and she essentially decided halfway through the summer that science wasn't for her. and so she stopped doing the project. So this isn't as strong. This is plagued by having small sample sizes. But better to learn that you're on the right thing. So what about temperature? Hot clear cuts, warm edges. The interior. Interiors are much cooler. So again, this just supports stuff that's been done before that shows that that edges, the clear cuts and edges are hotter and drier. And we know from the natural history of truffles that truffles don't like hot, they don't like dry. Okay, so then there's still lots of questions left in the whole story, but we've come a long way, I think, in understanding the response of this one species, redback voles, to forest fragmentation. And that is that it appears to be linked to its food source, truffles, and it's not a simple relationship with logs, so you couldn't just manage for redback voles or these other species by going out and cutting down a bunch of trees and creating more logs for them. It's more complex and probably mediated by abiotic conditions. So, okay, so here in the last 5 minutes, I just want to drive home just a couple more points. I don't want us to be fixated on just the response of redback voles, because there's lots of species out in the forest. And so one thing I want to just show you real quickly is to ask what response, you might quite rightly be saying, well, you showed us what happened to one species, but what about other species? What about other commonly caught species? Well, the answer to that is that their responses were really broad. And this is commonly caught on Oregon remnants. No, this is Dr. Seuss, because you always have to appeal to higher authority, Dr. Seuss on things. Dr. Seuss reminds us to think about the conditions under which an island, a patch of forest is really, that patches of forest aren't always islands. That patches of forest are really just islands if the species on them confront a hostile barrier. If it's hostile, if it's a hostile conditions outside of the remnant, then we expect the species to respond as if it's an island, but not if they're not hostile conditions. So let's look at some other species. These guys are really fascinating, and they're in the forest here on Mount Jumbo. Well, there's lots of different species of shrew. This is trowbridge shrew. They're endless entertainment. You can put them in a box like this and throw in anything alive. Grasshoppers, huge things, and they just run up and eat them. And you just sit there and you just say lots of thank yous that you weigh over 100 pounds and they weigh less than 10 grams, because if you weighed as much as them, you'd be history real fast. So what do trowbridge shrews do with this forest fragmentation? Well, they didn't show any response at all. So trowbridge shrews don't view this forest modification as being fragmentation. They're just as likely to be caught in the clear cuts as in the edge or in the remnant. Deer mice, Peromyscus, they love clear cuts. They love clear cuts, they love edges. So this is that good old-fashioned positive edge effect. Okay, some species show it, sure. And Townsend's chipmunk, by the way, another good fungivore, and we'll talk some about these guys on Saturday. Chipmunks and flying squirrels are very important fungivores, meaning they eat truffles. They show no edge effect, but they are less likely to be caught on the clear cut. Probably, again, probably linking back to the fact that their favorite food, one of their favorite foods, truffles, aren't in clear cuts. Okay. So this is just the bottom lines. Be careful about making predictions of extinctions or about predicting changes in forests, assuming that all the species are going to respond the same. They're not always going to respond the same. But sometimes creation of these really drastic effects can have big negative effects. And you can regard the third one. This is from another talk that I give where I talk about genetic effects of population isolation. And I assume that Mike Hillis is going to get into some of that stuff next week when he talks about things like corridors. I could go on for hours about that stuff too, corridors and connectivity and genetic stuff, but I won't. So Oh, this is just, you usually end a talk with sunsets, but this research is ongoing. So even though at the moment this truck is terribly stuck in the mud, this just shows that the research, it has some stopping points. It gets stuck sometimes, but it is going to get unstuck and continue on with future questions. So, so the last thing that I promised that I would do would be to say that, again, just to stress that the dynamics here are forest dynamics, but that there are some components of this which are in fact going to be likely to be found in the forest of Mount Jumbo, and others that are not. Like I say, Mount Jumbo wasn't just carved out of forest, but there a lot of the same kinds of species are going to be there. And so the very last transparency that I wanted to show you was one that was in a book, that was in an article published about flying squirrels, which certainly are up on Mount Jumbo. And the flying squirrel story is really quite similar to the vole story in the sense that, it just shows this remarkable connection and this remarkable importance of small mammals. Small mammals get a bad rap a lot of times. Rodents, of course, are 1/3 of all mammals, and of rodents, 2/3 of all rodents are microtine, mouse and rat-like rodents. But squirrels rest and reproduce in trees. They feed on truffles, hypogeosporocarps. Their fecal pellets contain the spores, nitrogen-fixing bacteria, and yeast. They deposit them on the forest floor. The spores germinate and establish new fungal colonies. The fungi, as we've talked about, get carbohydrates from the trees, and they return nutrients to the tree. And then if all is well and the fungi are hooked into trees, then the fungi accumulate nutrients and produce sporocarps, and all is well in the forest. But when you have small mammal communities that are disrupted, as foresters used to do intentionally, very intentionally, used to be drastic programs to kill small mammals, because the idea was that small mammals eat seeds, small mammals eat vegetation, small mammals sometimes eat little trees, and so there was this drastic campaign to kill trees. And what we're finding with this whole mycorrhizae story is that the small mammals are oftentimes really critical dispersers of these sporocarps, these mycorrhizal spores. That is an essential part of the whole forest picture, so that actually replanting or regeneration of trees may be aided by small mammals. So that's sort of just a little blurb on maybe some of the less appreciated dynamics of forests. And when we go out on Saturday, we can talk more about some of the specific dynamics that are going on with respect to these things. I don't know that we're going to dig for truffles. I've thought about it, but I don't know that we're going to dig for truffles. It's pretty destructive, digging for truffles. We might dig one or two random patches here and there and carefully cover them back, but you got to be a little careful digging for truffles.
Make it take a pig.
Yeah, I have this harebrained idea that I want to try sometime. I want to get a Townsend's chipmunk, like you saw there, and make a little harness for it and use it. Because Townsend's chipmunks are incredible at finding truffles. And I don't know where I'd put a pig, but a chipmunk you can keep in a little cage and have a trained chipmunk. But I'll let you know how that goes.
Fried chipmunks, fried truffles.
Yeah, and that was one other part of the red-back vole story that I didn't tell you in the analysis. Is we analyzed every trap where we caught a vole, and we asked, were truffles likely to have been dug up there? And it turns out that truffles were more significantly likely to be found where you caught a vole. So the two predicted each other. In other words, if you caught a vole there, then if you just dug right close to there, you'd find truffles usually. Or if you found a truffle and you put a trap there, you'd be likely to catch a vole. So.
Scott, how do you survey for truffles? Do you
Well,
just dig a certain section?
Yeah, the way you do it is, and this is the reason it's destructive, is you just randomly place down a bunch of different, usually people use 1 square meter plots, and you just dig down to the interface between the mineral soil and the humus layer, and then you dig down below that for just a couple of inches. And truffles are always found somewhere no deeper than, say, an inch or two below the mineral soil interface. So it's just a matter of digging up. So, however deep your humus layer is, 2, 4, 6 inches, something like that. And you just look for them, and they're, as you can see in that book, they look a lot like, what, rocks.
Yeah.
So a lot of times, you don't think you found one, but you have. But when you're in the forest, and we'll look at this on Saturday, keep your eyes open, because a lot of times you can see little digs. I'm sure you've seen this walking around in the forest. Little teeny holes in the forest, little teeny scuff marks in the forest, which can be lots of things, but a lot of times what that is small mammals that have dug up truffles in those little
Squirrels
scuff
all
marks.
the time.
Squirrels, yeah, squirrels are,
I thought they were.
flying squirrels are as mycophagous, that's the $5 word for it, eating truffles. Flying squirrels are as mycophagous as redback voles are. So, roughly 80% of a flying squirrel diet is truffles. And so, yeah, flying squirrels
What's
do
the
that for
conifers to the truffle like again?
Well, different species like different kinds. And you'll notice if you looked in that book, some of the descriptions will actually tell you what kind of trees to look under. So there are certain truffles that love oaks and certain love Doug fir, larch.
There's a lot of truffles up Eddie Canyon.
Yeah, sure. So, yeah.
I mean, is there areas that are more, or?
Well, moister areas will tend to be up to a point as long as it's not standing water moist. So a lot of times around here, north-facing slopes are good.
Yeah, I know they're a real delicacy to eating. I don't know if there's any poison ones.
There's not been any poisonous ones identified yet, but one of the cool things about truffles in North America is that they're finding new species every single year. And so the hardcore truffle people are always quick to say, well, there hasn't been any poisonous species found yet, but be careful, because two things. Number one, an amanita, a death cap mushroom that is not yet poked up out of the soil can look kind of like a truffle when it's just starting to come up, when the gills, when it hasn't opened up yet. So the truffle people say, number one, be careful and don't dig up a poisonous amanita that looks like a truffle. And number two, they say, hey, you might find an undiscovered species that we haven't figured out yet that might in fact be poisonous. So they do exercise caution about people eating it.
Larry Evans.
What's that?
Larry Evans, mycologist,
Yeah.
mushroom club.
Yeah.
We went up Patty Canyon and looked, but most all of the ones we found were already the chipmunks and voles and beat us to them.
Yeah.
Wasn't Larry trying to teach his dog to locate truffles?
Larry,
Yeah.
he knows farmers market.
Yeah, a lot of
sells
people-
mushrooms.
I don't see why not.
He knows where the mushrooms are. He will tell.
Yeah, a lot of
he-
people do talk about training dogs.
But do you know
Yeah.
if it can be
That's
done?
why I figure my chipmunk would be different.
Like your
I'll
drug
put it up
smell
on
like
the
dog
counter.
and mushrooms cool dog.
Yeah.
Is there a relationship to, if you cut down the trees, and those squirrels, like the flying squirrels depend on the trees, that's going to disrupt the whole cycle also, isn't it?
Yeah, right. And that's sort of a
Fragmented
more
out.
direct. That's right. That's a more direct effect. I was thinking I have to wind down here, but I was just going to launch into a whole other story because there's this. I almost, instead of doing redback voles, there's this amazing species that's in Washington and Oregon called the red tree vole, and this is a species that lives its entire life in the Douglas fir canopy. It almost never even touches the ground, which is remarkable because if you remember back from your high school or college physiology, you remember that pine needles are full of nasty terpenes and resins and stuff. Pine needles are not very digestible. Not very many things really eat pine needles. But these, and things that can eat pine needles typically are large bodies, so they have a big gut to process them. So for a small mammal to eat entirely Douglas fir needles is really remarkable. But anyway, if you cut down the trees, you'd obviously have an effect on red tree voles. So that's the obvious effect. The
But I
effect
mean,
on
and it's the whole cycle with the mycorrhizae.
yeah, right. And this effect with the truffles, the mycorrhizae probably stick it out in the clear cuts actually. They probably make it because they hook up with the alder and berries and stuff that comes up, new vegetation. But the species that depended on trees just don't have enough gusto anymore to make truffles. So what you see is lots of truffles for about the first 3 years after clear cutting, and then you see truffles go to near zero like we found until about 30 years, at least in Oregon, about 30 years after a clear cut, until the trees get reestablished again and get to be 10, 20 feet big, and then they can support truffles again.
Here, it might just stay gradually.
Here we don't know.
So slow,
Yeah.
dry.
I haven't talked to Larry Evans, for example, but I had a student summer before last do a little truffle study up at Lubrecht Forest up at Blackfoot. And she did find at least preliminary indications of the same kind of trends. She found lots of truffles in the interior of the forest, fewer at the edge, and even fewer in the clear cut. So it seems that the trend still holds. But she did find fewer truffles in general because it was drier. Okay, well, thanks.
Here.
See ya.
Thank you.